Hello, everyone, and thank you for joining us for today's webinar, AAV Analytics, Key Considerations when Developing a Clinical Program. I'm Heather Masaccio, your host from Cell and Gene Online and Life Science Connect. With me today is Will Fountain, Head of Analytical Development at Andelyn Biosciences. Over the next 45 minutes, he'll be discussing challenges that can stall an advanced therapies development. He'll share how Andelyn utilizes method validation within their processes and offers key considerations for taking your AAV gene therapy from concept to commercialization with advanced analytics. After his presentation, we'll be holding a live Q&A session. Feel free to submit questions as we go along. We'll get to as many as we can. If we don't get to yours, we'll follow up after the event. Just a few housekeeping reminders. If you run into any issues, please try refreshing your browser first. If you need additional support after, submit a question via the Q&A. Finally, the webinar will be available for on-demand viewing. You'll receive a link via email as soon as it's available. With all that said, I'm going to turn it over to Will to get us started. Will?
Welcome, everybody, and Heather, thank you for the introduction. As she said, my name is Will Fountain. I'm currently Head of Analytical Development at Andelyn Biosciences. I have about 2 to 2.5 decades of experience in the biopharma industry, initially starting with the University of Kentucky College of Pharmaceutical Science and Technology, moving on to Ventera Pharmaceuticals, Battelle Memorial Institute, and then joined Nationwide Children's Hospital in 2006, which then spun out into Andelyn, the company that I am representing today. I have a Bachelor's of Science in Chemistry, also from the University of Kentucky. So to give you a little bit of an overview of the topics we'll be covering today, I'll give you some introduction on Andelyn Biosciences, who we are, how we came to be, what we do. I will then move into adeno-associated virus specifically, giving some overview on that for those of you that may not be well versed in the virus, its use as a vector, and its biologics. From there, we'll get more deeply into AAV analytical considerations, talking about typical testing, focusing on determination of dose, comparability of orthogonal methods, genomic titer, genomic integrity, looking at AAV full and empty analysis, which is also very important, potency, host cell DNA, and strategies for qualification and validation. So to introduce you to Andelyn Biosciences, we first like to highlight the origin of our name. So Andelyn is a combination of two key patients in our history. Andrew was the first patient to receive a gene therapy treatment for Duchenne muscular therapy in 2006. Evelyn was one of the first patients to receive an experimental gene therapy for spinal muscular atrophy in 2015. That product is what ultimately became Zolgensma. Getting into our history a little bit, we're currently a full services CDMO with a long history in AAV. As I mentioned earlier, we spun out of Nationwide Children's Hospital. We have a strategic partnership and investment from the Cytiva Corporation. And today we have 3 facilities in Columbus, Ohio, in Central Ohio. In the last 2 years, we've had greater than 12 successful audits from clients. 2 of those included QP audits. We've been audited by the State of Ohio Board of Pharmacy for a dangerous drug license. And we've been successfully audited and a qualified supplier for NIH. And lastly, we have a couple of BLAs that are currently in preparation. Getting into a more detailed overview of our history, we date back to 1992 with work with AAV. That originated with the first AAV work in Dr. Phil Johnson's academic lab at Nationwide Children's Hospital. We then moved on into transient transfection of AAV, forming our original viral vector core in 1999. In 2006, when I joined the organization, that's when we were tasked with scaling up the production processes, making them GMP appropriate, and GMP appropriate analytics, and designing and building our first GMP manufacturing facility to create clinical grade vectors to enable human trials. From there, we moved on with production of cell banks, manufacturing of the clinical material that led to the Avexis Novartis Zolgensma product. We opened a second facility in 2017 that allowed us to have roughly 5 times the manufacturing and testing space, followed by further additions in capacity. And then in 2020, we formally spun out of Nationwide Children's Hospital as Andelyn Biosciences. As part of that spin out in 2021, we completed a strategic investment with Cytiva, part of the Danaher family of companies. That has enabled us to have close relationships with many different organizations providing manufacturing, purification, analytical, and laboratory instrument equipment and technologies. In 2022, we opened our 2 new purpose-built facilities that I'll discuss more later, completing with our grand opening in 2023. And now this year, we're actively working on PPQs for multiple different clients. Getting into our 3 facilities in a little bit greater detail. Firstly, we have our Andelyn Development Center, which is located in Dublin, Ohio, a suburb of Columbus. It focuses on process development, preclinical manufacturing, and analytical development. Secondly, we have our Andelyn Corporate Center located in Columbus. It focuses on warehousing, manufacturing, quality control, quality assurance, and other supporting departments within the organization. And then lastly, we have our Andelyn Plasmid Center. This was a repurposing of our prior GMP manufacturing space at Nationwide Children's Hospital, and now is primarily utilized to manufacture both preclinical and GMP grades of plasmids to further enable our raw material streams for AAV and other vector production. Getting into an AAV overview. So adeno-associated virus, for those of you that are not particularly familiar with it, is a 25 nanometer single-stranded DNA virus. It is non-enveloped with an icosahedral capsid comprised of 3 primary structural proteins, illustrated here as VP1, VP2, and VP3. The wild-type genome contains 2 genes, rep and cap, that are flanked by ITRs to create a 4.7 kilobase wild-type genome. Importantly, it can infect both dividing and non-dividing cells, making it very versatile in the use in gene therapy. And there are dozens of wild-type serotypes across both human and non-human primates that are utilized, as well as a growing list of engineered serotypes that can provide very specific and diverse tropism. The recombinant constructs come in both single-stranded and specifically engineered self-complementary AAV forms. And all of this together makes it a very attractive candidate for human gene therapy applications. To date, the FDA has commercially approved 5 recombinant AAV drug products, and we will go over those in a little detail. Getting into how AAV is produced, it is produced in vitro tissue culture. There are both mammalian and insect cell line production processes using both adherent and suspension cell lines. It typically requires 3 different sets of genetic elements. The first is the recombinant transgene cassette. The second is applying the rep and cap genes from AAV. And the third is helper virus genes. The viruses that are removed from the cells and purified extensively through downstream processes, ending with formulation. The graphic to the right illustrates 2 typical processes for manufacturing AAV. The first is triple transfection, where you have 3 separate plasmids, vector genome, AAV helper providing the rep and cap I mentioned, and an ad helper plasmid providing 3 adenoviral helper genes. These are combined together into an E1 expressing cell line, such as HEK cells, to produce the AAV virions. An alternative production pathway utilizes recombinant HSV vectors that contain the rep and cap or the gene of interest, which are combined together in suspension BHK cells and again make the same AAV viral construct. So it shows 2 different pathways to produce the same material. Getting into AAV purification, as I mentioned earlier, typically you either perform cell lysis or in many applications, media harvest of the AAV particles. You go through a clarification step. Typically there's a TFF step for volume reduction and or buffer exchange, followed by 1 or more of the following: ultracentrifugation, which can be either a step ultracentrifugation or isopycnic, various different affinity chromatography options, ion exchange chromatography, and typically TFF is again utilized to formulate the drug substance, lastly followed by sterile filtration to fill the drug product. The image to the right shows our FlexiPro purification system. We like to highlight this because uniquely it has a 100% disposable fluid flow path that prevents us from having to perform extensive cleaning validation studies, etc., all resulting in a greatly reduced chance of cross-contamination between different AAV vector productions. So moving on into specific analytical considerations for AAV, there's many different analytical testing stages requiring different levels of focus and different levels of development across the preclinical and clinical areas. There's also phased qualification and validation requirements across that clinical development spectrum, and a need to characterize products beyond what we normally consider of traditional pharma testing. So the first testing stage is raw material, focusing on things like cell lines, plasmid, production materials such as media, buffers, enzymes, purification resins, etc. We then move into in-process testing, looking at dose or concentration, safety, and purity as the product is both produced and purified into what becomes a drug substance or drug product, where we're typically looking at dose, identity, potency, purity, safety, and physical characterization. Lastly, we have stability, looking at both long-term frozen stability, short-term in-use stability, again, looking at dose, potency, purity, and physical characterization. And lastly, one area that is often overlooked is administration route comparability, or what we sometimes refer to as device compatibility testing. It's very important that we demonstrate that our infusion process, whether it be as simple as a syringe and a needle, or a complex infusion tubing with infusion pumps, maybe even as complicated as intracranial injections, are capable of delivering the vector to the site without absorbing, causing aggregation, etc. And so we typically assess that again through dose and potency. And another important factor to keep in mind is this needs to be proven for not just the human clinical device materials, but also any materials used to deliver vector in a formal preclinical toxicology study, if those are different than what is used in humans, so that you can show that the preclinical tox animals received the intended dose that you intend to give to humans. This is an example of a typical release testing panel. We have the traditional categories of dose, identity, potency, purity, safety, and physical characterization. Within dose, we have the areas of capsid titer and genomic titer, which we will discuss in more detail. Identity typically focuses on both DNA and protein identity. In potency, we have a spectrum of different types of tests, from transducing, or what's oftentimes called infectious, transcription, translation, or specific bioactivity of the product. Within purity, we have traditional protein purity, looking at the percent of full and AAV particles, which we'll discuss in more detail, along with genomic integrity, as well as a suite of residuals tests, testing for removal of process residuals, as well as others. Things such as endonucleases, bovine serum albumin, if the production process utilizes serum, host cell proteins, host cell DNA, surfactants, transfection agents, affinity ligands, ultracentrifugation gradients, if applicable, etc. Moving into safety, that tends to be a little bit more traditional with the areas of sterility, myco, in vitro viral contaminants, endotoxin, but more uniquely looking at replication competent AAV, which is an important safety parameter that must be evaluated in clinical applications. And then lastly, the physical characterization is relatively typical with pH, osmolality, appearance, aggregation, and subvisible particulates. Getting into definition of dose or strength in a little bit more detail, this is something that should be developed very early on in a development program to prevent additional bridging studies required later on. The dose is typically defined as a measure of the physical titer, and that can be broken into capsid titers and genomic titers. Different technologies support both classifications, and there are often significant bias and misalignment between the different technologies that we'll discuss in more detail. Importantly, PCR should be product specific. The FDA is accepting of non-product specific PCR, but they highly frown upon that and encourage people to develop product specific targets as early as practical in their application. And importantly, in the US, the FDA requires a dose determining assay, whichever one that is, be qualified prior to initiation of phase 1 clinical trials. So looking specifically at the categories of capsid and genomic titer, capsid typically will have multiple different types of units, such as viral particle, capsids per mil, or capsid particles. Supporting technologies can be ELISA, ion exchange, HPLC, multi-angle dynamic light scattering, protein to capsid conversions, UV absorbance, and we'll go into those in a little more detail. Importantly, genomic titers have a number of different possible units. Vector genomes, abbreviated as VG, genome copies, GC per mil, and an older terminology is DNase-resistant particle, or DRP per mil. These are supported by digital PCR, quantitative PCR, and in much older applications, Southern dot blot analysis. It's important to understand that all titering technologies are not created equal. They are not equivalent. There are significant biases between them. In this slide, I'm showing two different GFP control vectors that were produced. They were extensively characterized to use them as analytics controls in our facility. And so on the left side here, we have three different assessments of genomic titer, where you see the first area is qPCR with a supercoiled standard versus qPCR with a linearized standard. And you can see there are significant differences between those two measurements, but the relationship between vector 1 and vector 2 remains fairly similar. In the third area of digital PCR, we're using an AAV2 ITR generic PCR target. It is not specific to the transgene, but is a universal application. You can see that the relationship between vector 1 and vector 2 are almost identical, unlike the two qPCR measurements. When you move into physical titer, we have the UV extinction coefficient analysis here, where we're measuring the UV absorbance at 260 and 280 nanometers and utilizing an extinction coefficient to calculate the number of capsids per mil. In this case, you can see both the magnitude of the measurements are significantly different, but also the relationship between vector 1 and vector 2 is the opposite of what is shown in the qPCR application. Moving on to the right, we have an ELISA analysis, which again shows difference in magnitude and a difference in relationship between vector 1 and vector 2. And then in the second to last category, we have a protein assessment. So essentially, this uses the conversion factor of 1.5 times 10^11 capsid particles per microgram of total protein in a highly purified sample to calculate the number of capsids per mil. And you can see an opposite relationship between this value and the ELISA value. And then lastly, we have data generated from multi-angle dynamic light scattering, which again shows different magnitudes, but a similar relationship to the protein, opposite to the ELISA, but similar to the earlier qPCR-based and digital PCR-based analysis. All of this combined, it shows that no single assay is right, no single assay is wrong, and the importance of understanding the performance of a particular material across multiple orthogonal analyses to really understand the material that you're working with. Getting into genomic titer in a little bit better detail, the first area that I want to discuss is older technology at this point. It is based on quantitative PCR, which relies on a 100% efficiency to get an accurate measurement. You can see this demonstrated in this graph to the upper right, where the 100% accuracy curve is represented in blue with a slope of negative 3.32, and a greater slope at negative 3.8 shows both different values as well as an amplification of this difference as you get to higher CT values or lower concentrations on your standard curve. So this shows the importance of amplification efficiency in any analysis of unknowns. Traditionally, this qPCR was done using supercoiled plasmids as the standard in analyzing AAV samples. But over time, what we discovered was that the supercoiled state of the standard versus the linear state of the AAV DNA in the particles creates a bias because of the structural conformation difference and differences in amplification efficiency. This has historically shown a 2 to 8-fold higher titer when utilizing a supercoiled standard. So the next evolution was linearization of the plasmids, and that greatly helped in comparing linear plasmid standard curve to linear viral DNA. However, when you start talking about self-complementary AAV, it gets much more complicated based on the location of the PCR target. Essentially, the closer you are to the internal hairpin in a self-complementary construct, the more inhibited the PCR is in the viral DNA and lower the titer. So if you're looking at distal versus proximal, you're looking at up to a 10-fold difference in titer. All of this results in an inability to compare published titers. And I give an example where Hinderer in 2018 performed a large animal toxicology study utilizing doses that were published in the phase 1 clinical trial for what became the Zolgensma product. The study showed significant toxicology issues at that dose and raised great concern within the field about the safety of using these doses in patients. However, knowing what I knew about how that product was manufactured and tested, I was able to determine that Hinderer was comparing doses that were roughly 5-fold higher than what was used in that clinical trial because of the differences in their titering method versus the titering methods utilized in that clinical material and was cited in the clinical publications. So obviously that's a good example of how trying to compare titers or products manufactured or tested by different facilities, different analytics, can cause great issues in doing AAV research and development. So a newer technology for analyzing genomic titer relies on digital PCR, which is based on partitioning frequency and the use of Poisson statistics. So essentially, your sample of DNA, primers, and probe, and polymerase are partitioned either through droplets or, in some cases, drilled partitions in a chip into very, very small segregated reactions. They undergo thermocycling similar to qPCR, and any partition that contains the DNA of interest is amplified, the probe fluoresces, and creates an increase in fluorescence for those individual droplets. Any droplets that do not have that DNA of interest retain a low fluorescence and are able to be separated accordingly. So the technology is rapidly becoming the gold standard in the analysis of AAV. It holds a number of key benefits. It is an absolute quantitation. It is not dependent on the accuracy of preparation or the confirmation of your standard like the qPCR. It's also not sensitive to mild PCR inhibition. So it doesn't require 100% amplification and permits the use of general universal assays targeting the AAV2 ITR sequence located in all AAV products. And most importantly, it has greatly improved precision. We have been able to determine that digital PCR typically gives intermediate precision less than 3 to 4% for digital PCR, as opposed to the older 20+% oftentimes found with qPCR, which has been an important development in both satisfying FDA expectations for the precision in testing, as well as being able to compare doses, compare production processes, etc. There are 2 current obstacles that remain. To this date, there's really only 2 vendors that hold significant market share, providing instruments for this type of analysis. And in our experience, those pieces of equipment are not equivalent. One gives results that is approximately 5.1% different than the other. And it is not due to fundamental differences in technology, but is more related to determination of partition volume. So you can see here in this image to the right, where you've got different numbers of partitions with different amounts of target DNA that result in proportions being calculated, such as here of 70 positives out of 143 total. If the volume of those partitions has error in its determination, when you convert this proportion of positives to totals to copies per microliter or copies per mil, you're going to have some bias built in. An additional area of obstacle is there can be sample preparation bias when preparing the samples. And unfortunately, to date, there's no industry consensus on what is the appropriate way to prepare samples or how to deal with this bias when it does occur. How we get into that a little further. So essentially, there's 2 different ways you can prepare your sample. You can partition your AAV particles into the droplets or the holes in the chip, and then lyse the capsids, as is demonstrated here. So we have a tube with different empty and full AAV particles. We illustrate blue as being positive strands, red as being negative strands, and these go through partitioning, and each material ends up in a partition with some partitions remaining empty. We then lyse those capsids open and liberate that DNA as we do the PCR, and the end result is any partitions that had either a positive or a negative strand of DNA in one of those particles becomes positive and contributes to the calculated titer. However, if we start off with the same starting sample and we lyse those viral capsids, we release positive and negative strands of DNA into the solution, which may then anneal together. And when the partitioning occurs, you end up with fewer partitions with both a positive and a negative strand in the partition, resulting in a fewer number of positive partitions and a different titer calculated from that scenario. So one might think, well, that's simple. We can just multiply or divide by 2 to convert between those scenarios. However, it's far more complex than that. In a traditional single-stranded AAV vector, this is the situation we have where we've got a positive or a negative. We lyse it, and we have these 2 forms here, which can then anneal together into a single double-stranded DNA, or in some cases may remain in solution as single-stranded DNA, which can be dependent on slight differences in the ratios of positive and negative strands in the particles, or just inability of the DNA to anneal together. It gets further complicated when you start talking about self-complementary vectors, where again we have positive or negative strands of DNA. And when those capsids are lysed, they fold together into a self-complementary form, and the vast majority retain that conformation as shown here, but you can also have some of those unfold and anneal to each other as positive and negative strands, creating different ratios of annealed, or different ratios of the particles annealing together. So moving on into DNA identity by sequencing. That's an area of great focus right now. There are multiple different sequencing options, all with their own unique pros and cons. Within the area of next-gen sequencing, we primarily have Illumina, PacBio, and Oxford Nanopore technologies. And it's important to evaluate what is the goal of your DNA sequencing. Is it to prove identity? Is it to look at variants in your construct? Or is it to look at genomic integrity, which is a sub-area that is very hotly debated and analyzed right now? So looking at the 3 primary areas of Sanger, short read, and long read, you You can see that Sanger is a leader in terms of speed and low cost, but the short read and long read have a much greater ability to sequence through GC-rich regions or secondary structure. Oftentimes, when trying to sequence ITR sequences with Sanger, you are unable to get through more than 40 to 50% of the ITR sequence before the sequencing reactions fail, whereas short and long read technologies are much more successful in that area. The Sanger is also not able to detect low frequency variants. The way Sanger sequencing works, it's the strongest signal wins. And so if 40% of your molecules contain a variant, you may be blinded to that occurrence by the stronger signal from the 60% of molecules that contain the desired sequence. This is an area where short read and long read really shines in being able to get into very specific details of low frequency variants as low as fractions of a percent of the total sample. And long read technology is particularly suited for analyzing genomic integrity, which we'll get into in a few moments. Unfortunately, for all of the positives of short read and long read, they do not provide very clear black and white pass-fail reporting on sequencing results, which is where an area that Sanger does excel. So getting into genomic integrity, as I mentioned, it's a very frequently discussed issue right now. I've got a simplified example here of what a desired AAV transgene cassette might look like, where you have a promoter, a transgene, and a poly-A that is flanked by ITRs in the viral DNA. Unfortunat' we never have a sample that is homogeneous and represents this full form. We can have truncations from both the 5' or the 3' ends to varying degrees and varying extents. So this really matters quite a bit in the quality and efficacy of an AAV production. As you can imagine, this form here is our desired construct. If 99% of our particles are of this form where a portion of the ITR is clipped off, we may still have very good efficacy and potency compared to a form such as this where we're missing half of our transgene, where we would have very poor expression of the transgene, if at all. And then a final form that can occur in genomic integrity is where you have all or part of your desired genome, but also have a portion of the vector plasmid backbone, as illustrated here. There's multiple different ways that can analyze genomic integrity. The first is alkaline agarose jet electrophoresis. This is very good at separating single-stranded DNA by size. And you can utilize densitometry to get relative quantities of different populations. The limitation of it is poor resolution of similar sizes and a somewhat qualitative result with an inability to report a clear-cut pass-fail result in a GMP application. Another area that's been utilized is multiplex digital PCR. So in this case, we utilize different primer probe targets with different fluorophores on the different probes, as illustrated here in color 1, color 2, 3, and 4 across the length of the desired transgene cassette. Once narrow dilutions are performed to ensure that only one DNA strand is in each partition, you can look at the differences in titers derived from these 4 different targets to determine the percentage of fractions, the percentage of DNA strands that have all 4 targets versus maybe just 3, 2, or 1 target to get a assessment of genomic integrity. Lastly, we get into the long-read DNA sequencing that I mentioned earlier, where it is a very powerful technique. We get extremely detailed molecule-to-molecule variation analysis, where we can tell the difference between one strand and another strand that may only be one nucleotide difference in its truncation state. The limitations are complicated analysis, and again, the reporting is not conducive to a GMP lot release because there is a wide spectrum of different results, sizes, and impacts, and so it remains more of a characterization rather than a lot release. showing some specific examples of analysis of the genomic integrity, we go back to the GFP control vector that I mentioned earlier and look at some analysis of it. Importantly, you'll note that the viral DNA length is only 2,100 nucleotides in length, which is less than 50% of the packaging limit of AAV. And so we have to pay attention to the length of this viral DNA dimer length of 4,300. And so when you run an alkaline agarose gel, you can see 60% of our DNA present is of that monomer length and 40% is of the dimer. However, if you do packed biosequencing and analysis, we can get into, as a whole, what is the identity of all of the different strands sequenced. And you can see 94% were AAV forms in all their different iterations. 3% was other, so this would fall into categories like vector backbone. It would be the AAV helper plasmid, the ad helper plasmid, etc. We have roughly 1% that's host cell DNA, a very small fraction that's chimeric DNA, representing a single strand with multiple different identities, and roughly 1.5% unmapped. When you take this all AAV forms and you divide it up into which ones are single-stranded AAV and which ones are self-complementary AAV, which would be consistent with this dimer form up here, you can see a 45 to 54% relationship, which is the opposite of the 60 to 40%. Again, showing different technologies have different results and are not directly comparable. However, when you get into the specific genomic integrity analysis of this, we're taking these two populations and breaking them down into how many are full length, how many represent a left partial construct versus a right partial construct versus an internal partial containing no ITRs or containing backbone sequences. You can see the vast majority is full, but we do still have significant populations that are partials on the left and right with a very small internal partial and backbone populations. Unfortunately, in this analysis, you can't tell if that left partial is 99% of full length or if it's 10% of full length. And so it doesn't give a good analysis of the degree of truncation. So that requires further and more complex analysis to determine. Getting into another area that is hotly discussed is percentage of full AAV particles. So when AAV is produced, we get quantities of full, partial, and empty particles. These can be very difficult to separate from each other during purification due to their similar chemical properties. The issue becomes partials and empties can contribute to increased immune responses, but may not provide any or limited therapeutic benefit. And so we want to minimize them where possible. So again, there's many different technologies that can be used to assess this. We have analytical ultracentrifugation, which historically has been our gold standard. We have ion exchange HPLC, charge-coupled mass spec, mass spectrometry, and electron microscopy. All of these have pluses and minuses, as you might imagine. And to give some specific real-world examples, again, going back to that GFP control vector, we just talked about how it has a small size and pan-packaged dimers. And so here in this analytical ultracentrifugation analysis, you can see what the software is assessing as fulls is really only 13% of the total, but we can assume that is probably the dimers, where the partials, representing 83%, is actually the full construct. And then the number of empty particles is very small at 2.4%. Utilizing an HPLC-based approach, we are only able to discriminate between full and empty particles and have no visibility to the difference in full versus partial. But you can see here, two different purification processes result in highly enriched 97% full particles with very few empties versus a 33% full And so it's still very useful in assessing the performance of your purification process in getting rid of empty particles. Lastly, I'm going to show some mass spectrometry data, which if you compare the shape of the AUC curve to the shape of the mass spectrometry data, you see a lot of similarities, with shoulders on the left and right side representing the empty and the dimer populations respectively, with a large peak in the middle representing the partials or fulls, depending on how you want to look at this. But you can see that the software is quantifying both partials and fulls, or monomers and dimers in this application as all being full. So again, we get very similar assessments of fulls and empties, but limited ability to separate and quantify partials separate from the others. Another area of importance is potency. There's many different approaches to potency, and many of those approaches suffer from poor precision in testing. There's a spectrum of development in potency testing, starting with preclinical studies, and it's important to establish your potency reference standard as soon as practical in the development of a clinical program, so that you have a consistent material to compare all future lots to throughout your development phase. It's also important to have a quantitative assessment of biopotency going into your phase 1 clinical trial, and then have that qualified as you're going into phase 3 trials. Looking at the different types of potency, there's transducing titer, which oftentimes is referred to as an infectious titer and is analyzed by TCID50 on a RepCap expressing cell line. You can also have measurements of transcription, translation, and functional potency or bioactivity using the mechanism of action of the AAV drug product. So showing two specific examples, the image here to the top right is an example of how a transducing titer might be performed, where it's in a 96-well format, and we're scoring individual replicates at individual dilutions as either positive or negative for amplification of the viral DNA to be used in a Spearman-Carber equation to calculate infectious titer. To the lower right might be utilized in an ELISA, where we're looking at expression of protein in tissue culture, et cetera, looking at the performance of a test material versus a potency standard, and are able to calculate the difference in relative potency between the two. One of the last areas I'll be covering is host cell DNA. That's another area that's received recent discussion and interest. There is a WHO guideline for host cell DNA in viral vaccines that calls for less than 10 nanograms of DNA per dose and less than 200 base pairs in length. We suffer from a lot of challenges meeting this guideline in the area of AAV gene therapy for a number of reasons. Gene therapy is inherently different than vaccines. The routes of administration are different. The volumes of drug administered are oftentimes much different. And the concentrations can vary considerably. Some programs have very large systemic doses where you may be infusing 10, 20, 60, 80 mils of drug product intravenously into a patient, which is obviously much different than, say, a half mil vaccine being delivered intramuscularly. And encapsulated host cell DNA is unable to be removed. It's a full viral particle with DNA in it, just like the desired product, and therefore it's very challenging to separate. And you can also have packaging of certain chromosomes at higher frequencies than others that can bias results depending on what your target is when assessing host cell DNA. So there's a number of different approaches for how do we quantify this. There's commercial kits versus proprietary assays. There's qPCR versus digital PCR. You can look at human genes versus adenoviral genes, such as E1A, E1B, that's in the HEK host cell production line. You can look at different amplicon sizes versus a single amplicon. And there's also the possibility of using next-gen sequencing, to look at the identity of strands of DNA and do they map to the host or to the viral DNA. So I've got some examples here, again going back to that GFP control vector that we've been discussing, where we're targeting the human RPLP0 or the AD5E1A genes with amplicons of roughly 200 base pairs in size to be consistent with this less than 200 base pair assessment from the WHO. Then we're able to get total DNA in nanogram per mil of 215 or 194. Obviously, depending on the dose volume, that may be much greater than this 10 nanograms. However, when we look at the encapsulated DNA, we can see that there's very little free DNA present, showing support for the statement that the encapsulated host cell DNA is unable to be removed. So there's really not much that can be done on the purification process to improve these numbers. Through careful design of experiments in developing production conditions, we can drive these numbers down, but once the particles are produced, there's little that can be done about it. When you compare this to the genomic titer, the molecular weight, we can calculate the total amount of viral DNA of the DNA of interest, and we're able to get assessments of what is the relative percentage of this host cell DNA in this product. And so you can see that there is 2.1% and 1.9%. On our PacBio analysis that we spoke about earlier, we had shown that 0.9% or 0.96% of the reads mapped to the human genome. And so this shows that two completely different technologies, one containing two different targets, can give very similar results in the overall assessment of host cell DNA. Lastly, I want to get into qualification and validation strategies. So it's important to have appropriate development, qualification, and validation at relevant steps in the developmental pathway. So we have preclinical, phase 1, phase 2, phase 3, and commercial as our primary stepping stones through the development and validation process. Our recommendations are to have genomic titer developed for your preclinical studies. As I mentioned earlier, it should be transgene specific so that it can identify the specific viral vector that you're working with. And then as you go into phase 1, phase 2, it should be qualified, which does not define acceptance criteria. Moving into later phase, you use the qualification results to define acceptance criteria and really drive the conditions under which validation is performed. When you get into infectious titer testing, it's important to show method feasibility and show that the genomic titer primers and probes are suitable, but we don't necessarily need to go into extensive development because it is a platform asset. Qualification and validation typically is not required for phase 1, 2, but is for late phase development. Other assays that are non-compendial, the development needs vary considerably program to program and based on client desires. And so we can develop, qualify or validate, and finally validate as appropriate to meet our client's needs. And so you should consider what is the appropriate timing for your application. Do you want to have validation done early, or do you want to do minimal qualification validation work until after efficacy has been shown in the phase 1, 2 trial? And then lastly, with compendial assays, we typically recommend doing qualification or verification as part of a late phase activity. So to summarize some of what we've talked about today, there's different types of titer. There's physical, genomic, infectious. The terminology of each is important. The chosen technology for each is important. And there's no true right and wrong answers. It's much more of a spectrum of what does this product perform like in different orthogonal tests. So again, technology is super important in what you choose, how you use it to minimize error and variability. Selecting a PCR target is of critical importance. I talked some about the AAV2 ITR target being universal. That's great in early discovery science work, but we really want to move towards something that is transgene specific as soon as possible and avoid using promoters or common poly-A's when possible. You also want to consider the host cell DNA cross-reactivity when choosing a PCR target. If your transgene is a native non-codon optimized human gene, then when you're doing infectious titer testing on a human cell line, you're going to get cross-reactivity and an inability to measure your amplified viral DNA. Again, a good quantitative biopotency assay is invaluable as early as possible in development. And it's extremely important to understand your analytical bias and be consistent. I like to say that it's better to be consistent throughout the lifespan of a program than be absolutely accurate in your analysis, because we've shown the different technologies all have different accuracies. And so that becomes less important than high precision and consistency within a technology. Again, importance of establishing references as early as practical, so you have a common material that can be your baseline in comparing different production facilities, different production processes, lot-to-lot variability, etc., and being confident that it's not analytical variability by the use of your analytical reference. Again, to do that, save retains. You can never save too many retains from too many early productions, so that you can do comparability should you have to switch manufacturing facilities, etc. Those materials end up being worth their weight in gold, or probably much more than their weight in gold, if it comes time to do comparability studies. And also begin stability evaluations early in development. It's always important to be able to demonstrate that vector going into a preclinical toxicology study is stable in the conditions that you're storing it in, or it's stable in the in-use conditions in that toxicology facility. And lastly, don't forget to study the delivery device, whether it's being delivered in humans, non-human primates, small animal models, etc. Being able to demonstrate that you're able to deliver the desired dose is very important in both preclinical and in IND filings. So with that, I'd like to thank everybody for attending today, and we will switch over to the Q&A portion of this webinar. Thank you, everyone.
What a great presentation. Thank you so much, Will. Let's get right to the questions, audience members. Just a few quick reminders before we get into those. On your screen, you'll see a Q&A section where you can ask your questions. And there is a speaker bio section with contact information should you want to reach out for more information later. Okay, let's go to our first question. Okay, Will, how do choices you make for analytical methods impact later development as you move through clinical trial phases? What considerations do you recommend?
Yeah, so thank you. I'm going to answer that in the context of genomic titering methods and the importance of establishing that technology as you're going into preclinical toxicology testing and sticking with that. It's not essential that you have a transgene specific assay going into the transgene toxicology, but it certainly helps eliminate additional bridging studies down the road. And so you really want to establish a strategy early on and stick with it, but don't get paralyzed by indecision if you're stuck between two things, because you can change strategy down the road. It just requires additional bridging study to do so.
Okay, very good. Go on to our next question. In your opinion, can dPCR and mass spectrometry be run in a GMP QC setting?
So we're still figuring that out. We are absolutely using digital droplet digital PCR in GMP settings at this point. We are currently utilizing digital PCR, not the droplet form, in our R&D laboratory, in R&D applications, and transitioning to that GMP space. both technologies have pluses and minuses, and we're still working on that GMP implementation for that. For mass spectrometry, that's still fairly new to us. It's something we're still evaluating. I think a lot is going to depend on the validation of the algorithms used to differentiate between fulls and empties, and what kind of accuracy and resolution it can get there to determine if it's going to be a true GMP appropriate method or not.
Okay, thank you. So looking at our next question, when many methods give different titers of the material, how do you confirm which is the correct one?
There isn't really a correct one. The correct one is the one that you're using. As I've shown, many of the different technologies disagree with each other. The important thing is to pick one and stick with it. Consistency is much more important, but you should understand the other assays, what results they give, and understand how you may or may not be biasing your results compared to the results of others based on the assay that you chose. But there really isn't a gold standard to date on what's absolutely most accurate or best to use.
Okay, thank you. Now, Hazel wants to know, do you have a preference for qPCR or ddPCR for HC-DNA?
Yeah, so my general approach is the digital PCR is better. It doesn't require maintenance and control of standard material. It has much greater precision, and so you're not wondering about the differences in residual host cell DNA between two lots. You know, is that just variability of the assay, or is it a true difference in your purification process? So I think digital PCR wins hands down there. The one thing to consider is that if you do have very long strands of host cell DNA, it may impact the partitioning of the sample and create some issues there where you would not experience that in traditional qPCR.
Thank you. So I'm just checking out the next questions coming up here. And the next question is, let's see here. Is AAV biopotency required to initiate new IND when it is used as a vector for CAR-T cell?
Yeah, so that's an area we have some experience, not a lot. Most of our clients traditionally have used the AAV directly in vivo. So it's not an area we've seen a lot of. And in general, biopotency should be developed as early as possible and be as quantitative as possible. But the FDA and other regulatory agencies do understand that it's very complex. Sometimes it's not easy to get a quantitative result. And so they're generally understanding of that and allow a spectrum of development in that area.
Okay, excellent. I think we have time for one more question. Let's see here. Okay, well, for residual host cell DNA, Will refers to the WHO guideline, yet the final FDA guidance for neurodegenerative diseases dropped this limit. Do you think that the WHO limit is a concern for countries other than the US?
That's a little bit remains to be seen. I think a lot of people understand that trying to apply that WHO limit to gene therapy applications is a little bit of a square peg in a round hole. They understand, doses vary wildly, routes of administration vary wildly. Gene therapies are, delivered once and done. It's not a case of, annual or multiple times a year vaccines, those types of things. So while the FDA has come on record and kind of said, we understand that this is an unreasonable limit, I think many of the other agencies will follow, but everybody's still going to want controls on host cell DNA, keep it as minimal as possible. And it's not just a case of, You can administer whatever you have. There's still a need to control that there.
Thank you so much. I think that wraps up our time today. Many thanks to our knowledgeable presenter for sharing his time and information with us, and to you, the audience, for joining us. Remember, if we didn't get to your question, we will follow up with it after the event via email. Have a great rest of your day, and we'll see you next time.
Lovely.